All right, everybody, let's go ahead and get things rolling. Thanks for braving the weather and the security guards to be with us this evening. Glad to see all the folks in person, and thanks for the people that braved the trip to your couch to log in online to watch the live stream on Zoom. Just before we begin, just a couple of quick housekeeping notes. So we are accepting donations now. So all the food and beer is really, I'd like to thank our supporters. So we have our first one, Nexomics Biosciences, is tax deductible. So we are incorporated with OMSF, so it's charitable. So any folks out there in biotech or industry, pharma, you can hit this little QR code here and go on and donate. If you have any questions, also please feel free to reach out to me. You can just email bpdmc@omsf.io. That goes right to me. Great. And the other point I'd just like to say is we've got some exciting speakers lined up, but we still have openings for the rest of 2024. So also on our website, we've got a nomination form where you can nominate speakers. Feel free to nominate anybody from anywhere in the world, but know that we probably are only going to invite people that are local in Boston, or maybe while they're in town. If we do really stellar with our fundraising for donations, we might be able to fly in a speaker, but for the most part, we'll be local folks. We also accept self-nominations, and we have speakers from any career stage. So if something is cool that's going on, we would love to have that person present here. All right. So I'm very excited for our speaker today, Rubul. So he did his PhD actually in Massachusetts on the western side in Amherst. And then for postdoctoral fellowship, moved out to Seattle, where we met at the Institute for Protein Design, and then came back eastward as a research fellow now at Boston Children's Hospital, and very excited to see what you've been up to. So please take it away.
Can you hear me? All right. Thank you so much, everybody coming. And like Chris said, the weather is so bad, but still, the good turnaround. That's great. So I want to start by thanking Chris and Sergey. Where is Sergey gone? Oh, right there. Sergey and Nick setting up this platform. This is really wonderful platform. I've been here multiple times in the past, and great talks always, and from all different career. Different stages of career. So this is really wonderful platform, and I thank them for having me here today. So today I'm going to talk about. So this is a story. It's about designing protein and really using them for like cellular engineering, like stem cell engineering and things like that. And this story, it was done both at IPD at University of Washington as well as here at HMS. And so basically, I'm going to like talk about both the protein design problems and what I have been doing, and then how I've been using that for engineering cells. I'm always inspired by how cells interact. So when cell interacts, actually, there are a lot of things going on, signaling between cells. And one particular example is the immune cells. When they interact, they actually form this nice, what is called immunological synapse. For example, here, the T cells and antigen-presenting cells, they interact through this immunological synapse, where there's not just one molecule doing signaling. There are lots of molecules that come together to interact and really execute the signal. And finally, you will have an outcome, right? And so that, if you zoom a little bit, this is very crowded slide, and which I did not make. I took it from internet. But as you can see that there are lots of signaling molecule interacting to each other. In this case, this is a T cell and then antigen-presenting cells. And so you have TCRs, MAC, then you have all the stimulatory and co-stimulatory signals like CD4, then you have signals like CTLA4, then PD1 and whatnot, right? So all of these molecules, they come to this junction, the cell-cell interaction junction, and it actually forms a what you can call it a signaling hub. it's basically, it's a lot of, molecules coming together to act together. And so this is not only in T cells and antigen presenting cells. It happens between T cells and target cells and all different kind of immune cells that actually do cell-cell interactions. So that's really inspiring for me as a protein designer. And then the other piece of inspiration that I draw is that the intracellular signaling hub. And again, there's not just one single example. There are like so many, the machinery, cell machinery, actually they all come together to do something important. And for example, here, this is a apoptosome, which execute the apoptosis. And you have, FF1 and the cytochrome C coming together to make this assembly, and which actually executes the entire, the apoptosis pathway. And likewise, the inflammasome, who is also in response to inflammations, lots of the 3 different proteins come together to build these assemblies, and they execute a particular signal. So when I, a couple of years ago, when I went to University of Washington to work with David, so I had a really ambitious goal. I wanted to build something totally from scratch, like totally de novo. I wanted to build some kind of signaling hub, which can be then used for many purposes. For example, you can actually control, you can bring different signaling molecules together, and you can control the signals. And you can also, like, not only like what is known, but also you can bring unknown signals, and probably they never, got together in nature, but you can bring signaling molecules together, and then you can ask questions, can they work in some meaningful way? So that was my very ambitious goal. However, in order to build this kind of signaling platform, a signaling hub, you need a lot of building blocks, a lot of units. For example, here you need a homohexamer. This is a hexameric proteins. You need oligomeric, you need, for example, heterodimer here. You may need a heterotrimer here. Then it goes out to another heterodimer. Then it comes back. And then if you enforce symmetry, then it should be able to make the entire thing. So again, this is in picture. It is very simple. However, to build the entire thing de novo from scratch, it's just one of the outstanding challenges that we could not have think about a couple of years ago. And so that is one problem, that not having enough of this building block to build this is one problem. The other problem was that even if you have these building blocks, like hexamer, heterodimer, heterotimer, you would also need to connect them through sort of like really rigid arms, because you can't have flexible linkers here, because then you will lose the symmetry, right? Things will start flopping around. So you will not have a very rigid structure. So you would also need this long extending arm. So for example, here from this heterotrimer, this arms coming off, and then you have a heterodimer. Similarly, you have arms. So that was where I started, and then obviously we realized we can't do this, and so we actually went back to the board. And one of my early days there, a lot of people like David and Will Scheffler, Jon Shia, and TJ Brunet. Many of you might know these people. And so we sat down and we started talking about how we can actually build this. Is it even possible to build that? And so we started with very simple structures. Let's not get into really complicated stuff. Let's start very basic. So for example, here you can do a dimer, and then 4 dimers make a tetramer of a dimer, and so that can be built. And then you can take this, and then you can further install heterodimer in between the dimer. Then you can even extend it. You can have a bigger structure, right? So that way you can keep going, and then you can really make it. So we had a lot of this brainstorming session, and we talked about what we can do and what we cannot do. So anyway, so now what I'm going to do today is that so from that very early meeting, how far I have gone, what did I actually build, and like my main goal that trying to use this as a signaling hub can I actually use them, and what kind of meaningful things that I can do. So what I'm going to do is that I'm going to present the designs that I made, and then I'll talk about how I can use these designs for building signaling hub, and then finally talk about how we can actually engineer cell-cell interactions using these protein molecules. So going back to the problem of extending arms, and so what can you do? So you can take, for example, a homopentamer. This is a pentamer. This is called C5. C5 is cyclic 5. That's a pentamer. I mean, you were seeing from the side, so that's why you don't see well. But this is a self-assembly of pentamer. So you can take one of these helical bundle pentamer. Then you can take a repeat protein here. This is called design helical repeat protein. This was made by TJ Brunet, one of the members. And so what you can do is that you can bring them together, and you can actually superimpose one of the helix from the pentamer and one of the helix from the repeat protein. You can superimpose, and then you can make a chimeric protein like that. So it's basically just trying to superimpose the helix and then rebuilding the, wherever you are basically, redesign that with Rosetta. So we used to use Rosetta to redesign protein sequence. So, and then, so if you do that, then you have this pentamer at the core. Now you have these arms going out from there, because then you have, you just basically fuse there. And so we built a protocol, and that's called helix fusion. And this was actually written by TJ Brunet. And then once you have this building block, once you have enough of this, like, let's say homodimer, homotrimer, things like that, you can take those, and then you can further reinforce symmetry. And so what happens is that, so let's say you take a homotrimer with an extended arm, then you have a tetramer with an extended arm, then you can put this in a certain angle, and any missing piece, you can again put one of this repeat protein and just try to stitch that, the whole, whatever there is. And so once you do that, then if you enforce symmetry, then you will be getting all the, symmetrical big, large protein complexes. So the entire protocol then breaks down to 2 steps. In the first step, you basically just do helical fusion. And in the second step, we call it worms, because these are all looks squiggly worms. And so then you can use symmetry and use the helical fusion protocol to build large assemblies. So with these 2 protocols, so we started designing proteins very rapidly. And so ideally what we do is that we design these proteins in computer. And then once we are confident enough, then we order those genes, and then we express and purify the protein, then pass through any separation and purification columns, and then finally we do structural characterization, where electron microscopy or crystallography. So these are set of those the proteins that we built. So we started with the core tetramer here, and then we extended with the arms that I mentioned. We used that fusion protocol. And so this is a tetramer, and this, you can see the cryo-EM picture here, and nicely, looking exactly same with what we designed. So likewise, we have pentamers here, and then more pentamer here. This is negative staining electron microscopy picture. We have hexamer. This was actually very interesting. I really liked it because it looks like a flower vase, And then this one is again another hexamer. This one was also very interesting. This looks like the inflammasome, the complex that I showed. It looks very similar to like that. But this is only hexamer, and this is it is not composed of many other proteins. So then,
are you going to comment on the design goals that went into those secondary structures? It looks like that you used and picked for this. It looks like very repetitive secondary structure.
So yeah, so I can I did not include that, but so it begins with so helical bundle, like the core begins with only helical bundle, like it's a 2 helix, hairpins. And then so those are self-assembled already through docking and things like that. And then the arms that I talked about, so those are called design helical repeats. So those are basically helical repeats, like hairpin, 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 hairpin. And you can increase that length. We have, from like, let's say 2 hairpin to all the way to, you can make 10 hairpins. So those are again very modular modular hairpin structures. Yeah, so that's the structure that we started with. So then once we had this, the first layer of designs where we have all these oligomeric proteins with this extended arm, then we started worms where you will be now enforcing symmetry to basically build further bigger structures. And so, for example, here, this is a dimer with these arms, and then this is a heterodimer with arms. Then there's another homodimer with arms. And if you enforce symmetry, now you can have this homodimer at the opposite sides, and then heterodimer basically just splicing in. And so you can build this dihedral protein complex, which has basically 8 chains. I mean, here's the model. I mean, it looks very simple here, but it is actually made of 8 chains of protein. And so when we did the structural characterization, so you can see that it's nicely overlaid with the 3D reconstruction of the model. So then we.
Confirmation they assume, or do they also get the different?
So that's a very good question. So usually what happens is that we have in our design pipeline, we would have, let's say, out of like 50 designs, we pass through, different filters, and then we'll select 10 out of that, and then we'll do all the characterization. Some of those we do see alternate conformation. I actually ran into a very interesting conformation that actually switched, but very soon I also ran into a rabbit hole. I could not basically, so I gave up on that. So there are cases like when you actually get to alternate conformations and things like that. So here again, this is going to be my last set of proteins, I promise you. But so then here again, this is 5 heterodimeric proteins self-assembled using the worms, and so you can build this very nice structure. So we call this one crown because it just looks like a crown. And then we have tetrahedral symmetry where there are 6 homodimer and 4 homotrimer, and then it builds into a tetrahedron case-like structures. And likewise, this is an icosahedron case where there are 30 dimers and 20 trimers, and it all builds into a tetrahedron.
Can you talk about yield
of the protein?
It's all produced in the same bacteria.
So
they assemble in or pure.
That's a very good question. So most of these are one component we call, because they're one chain, but it repeats. So because it's only one chain and self-assemble, when you actually express in bacteria, they're going to self-assemble. However, there are designs when
What's the yield?
this yield,
How many, when you look in the EM, how many disassembled structures do you see?
so what we do actually, after purifying, we run through SCC. So when you run through SCC is size exclusion chromatography, then we do see some misfolded thing, but we pick the one that is like really good, very high percentage of basically right size in the SCC, and we take that one to our EM. So what happens is that we already pre-select the good ones, right? And then bad one we hardly look at. And bad one, like there may be some fraction of it. And that design to design, it varies. So usually the design that have a lot of misfolded or something like that, which is not coming in the right size, we tend to disregard. These ones? So that's another great question. So these are because these are de novo proteins, and they're made of helical bundles, and they're designed in a way that they're very stable. So if you boil them, even at 90 degrees Celsius, nothing happens. They're very stable. So if you have to disassemble, the only way is to find some detergent which can break it. And many of these are very, they are like hydrophobic interface. So it's very difficult to break those. However, we had designed where we had hydrogen bonding in between. And for those cases, you can change the pH. And I think a couple of months ago, Erin gave a talk about using pH to disassemble them and things like that. So yeah, there are different designs that you can use that. And then people also use disulfide, for example. I think you work on disulfide, right? And so there are other ways to sort of design the interface so that you can treat them. Yes, sir. Yeah.
So the tetrahedral and the icosahedral are both using C2 and C.
That's correct. C2.
Do you get mixes of them, or do you only get them?
So both the origin, the building blocks, initial building block might be different. They are not started with the same building block. So ideally what happens is that you will get the right one. If either you get the right one or you don't get at all. You usually don't get like, you're designing icosahedron and you will get tetrahedron. Usually don't happen.
So you start with the building block.
That's correct. That's correct. And many times actually it fails in the building block level itself, because some of the building blocks do not have the right geometry and shape and size to fit into whatever the structures that you're trying to build. So yeah. So anyway, so these are some of the designs that we work on, and we published about 2 years ago, and we actually had lots of other designs that all like homo oligomers, and then using worms, even making higher order protein assemblies. And so these were then these were the basic protein structures that I use for my what I proposed initially, that I want to build the signaling hubs so that you can do very interesting things. So so one of the, as I said beginning, that there are a lot of intracellular signaling hubs, and one of the signaling like whatever that recently people have focused a lot is the intracellular condensate, right? And in last decade, people have been exploring so much about how proteins actually come together, and then they actually they gather together and do something interesting, right? And if they're diffuse all around us, all across the cells, they can do, but when they come together, concentrate, and they can do stuff. And so that's kind of, again, trying to bring all the molecules together in proximity so that you can do some signaling. And so these granules are made of proteins that have disordered domains, that have like some floppiness in the protein structure, not only very rigid protein, but some floppiness, so that you can have the long range the protein networks. So I was very interested in this, because if you can build this long range protein network and then let it assemble inside the cells, then you can actually do new functions inside the cells. So so in order to do that, like I said, you have to bring some kind of disorderedness into this protein. You can't really do with pure structural protein. I mean, you might be able to, but in different ways. So what I wanted to do that, for example, I have a pentamer here, and then and because I want to basically put some disorderness here, so I can put a floppy linker here, and then put a interaction moiety domain here, and which then I can assemble this one to a dimer, and which dimer will also have some kind of the flexibility in the structure, and then when they assemble, then they will just assemble randomly and will make a very extended protein networks. And so before we would, if we have to do this for the ordered structures, we will use symmetry to tightly enforce things. But here we do not need symmetry because it's kind of symmetry breaking. So, and the kind of chemistry that we use to assemble this protein is either covalent, where you can take SpiTag and SpiCatcher, which is this is a lot of people use for conjugations. This is like just like a click chemistry. And so that's covalent, but also you can use non-covalent, where you can use designed heterodimer. These are heterodimers that are de novo heterodimers that are built in the lab. And so what this allows is that it allows the dynamic interaction, basically on and off kind of stuff. And you can really control things in dynamics and things like that. So so what I did was that, so I took this pentamer, and then so then another thing about this condensate is that they also have a mechanical strength within it, and that mechanical force actually it eats up one more layer of signal, because there are a lot of signaling pathways that actually require mechanical stiffness of the material, like both in extracellular as well as intracellular. So so another thing that I wanted to incorporate to these proteins is that the mechanical strength that will then guide certain signalings within this the protein complexes. And so in order to do that, so what I did is that I basically varied the flexibility of the linker from, for example, here I have a GGS block, and then I have here 5 GGS block. This 5 means 5 GGS block. And then I have, for example, 10 GGS block. And then by varying that, I wanted to see how that basically gives me differential kind of mechanical strength of these protein assemblies. And then, so then I would assemble this in test tube. I will purify, express and purify both separately, and then assemble. And you can see this beautiful, hydrogel looking material, which obviously eventually we want to do inside the cell, but just to characterize. So we made outside and then we build. And then when you looked at the stiffness of these proteins, so this is a measure of stiffness, and we measure by something called Storrs modulus, but I'm not going into details. But you can just see that the stiffness of these protein complexes actually go up from the short, the flexible linker to long flexible linker. So when you have long flexible linker, so the stiffness of these protein complexes is much higher than the short one. Yes.
Did you find that counterintuitive? The longer.
So yeah, that's a very good one. That's what came to my mind as well. And so, but actually, we ended up doing a lot of molecular dynamic simulations, and what we found is that with short one, there are like a lot of holes available, and they can't really extend out very far, and so that actually makes they lose their stiffness. Yeah, this is a very good question. So so that was so this one with flexible linker. And so we also wanted to explore with sort of rigid linker land, because as I said, yes, question. So these are all so usually when I do experiments, so I always try to use PBS because that's more relevant to the cells when I do.
So are these
These are not, because there's no hydrogen bond network. Yeah. So so then I also then changed the length of arms. And then wanted to see how that actually impacts on the mechanical properties of these protein assemblies. And so here, keeping keeping the linker floppy same, but extending this the rigidity of these arms. And what we found is that again, this the stiffness plot here, you can see that with longer arms, so you see basically the stiffer materials here again, just as that we saw in the flexible linkers. So so then after we yes, first.
Before you move on to the gels, how concentrated were the proteins?
That's very good question again. So usually we try to make because we are comparing from each other, so we try to fix that, and we usually do 10% width by volume. However, we can go down to like 0.5%. It's fine too. But usually we standardize at 10% so that we have the same amount of material. So so then question no.
Why don't you use molarity?
Molarity?
Because that is sizes, right?
That's yeah. So we also tried with molarity, but at the end we can't change both. So it's very difficult. Either you have to have the same density of material, or you have to have basically it's very difficult to change two parameters. So we tried with molarity as well, but I think the density was more kind of with molarity there's some kind of variability in terms of how much you have material there, because with smaller molecule there can be a very less amount of proteins. So so then finally we move on to the cells, and we try to express these to proteins. And again, as you can see that with the shorter the arm length, you don't see much of this condensate formation. However, when you go extend the length of the arms, so you see more and more condensate formations, and you can actually measure the number of condensates that's being formed here, and as you can see that with the longer arms you have more condensate formation here. Yeah, that's a question. So that's a very good question. Because it's a binary, we are on we have to co-express two things. We mostly looked at, we never purified them and then try to see them what is there. But it's mostly, I mean, these proteins mostly there. I mean, when you purify, we don't really see any other protein coming along with them. So I'm hoping that they're not interacting with any other protein. But if that does, I don't know that. So so then so now we characterize the stiffness in outside, but to characterize the stiffness inside is again another problem. And how are we going to measure how stiff the material is? So what we did is that we did something called FRAP, which is many of you might know. This is a fluorescent recovery after photobleaching. So what happens is that so when you have this the condensate, so you shine laser there, and so when you shine laser, you bleach out the color, and then when you remove the laser, so then you look at how much the diffusion happening back to that wherever you shine laser. Basically, you're trying to see the recovery, how fast this material, this color move back to where you have a bleach. And so depending on how fast it moves, you can predict how stiff the material could be. So if it is like really tight gel-like material, then most likely that things are not moving back, and it's going to take a very long time. So what we found is that with the long extended the arm length, the things are not moving back because there's no recovery, and that means it's very stiff. Versus as the length, the arm length, becomes smaller, so then you can see that the recovery is higher, meaning that you have basically softer materials. Yes.
Got a question from the folks that are on the live stream. Philip asked, "Did intracellular condensates form at lower expression levels?"
So the question is the intracellular, extracellular? Intracellular condensate. So we did not have a control for so we had like regular CMP promoters, and then we just let it express. And I think at some saturation, so they just start making. So I don't think we have a proper control for how much we are expressing versus. But they are we try to because it is involved with two plasmids because there are two components. So we try to make sure that they are getting equal. So yeah. So all of this question.
Eventually they will be
So it's it's hard to say. It's also depend how long you are expressing. If you are expressing too much, I think it could be also toxic. So I think there needs to be some kind of control how much you are expressing. But in long run, things like that can happen too. Yeah. So so the data that I saw, it was mostly through this the covalent interaction between this protein, right? The spike and spike catcher. So then and that is very irreversible interaction. So what happens if you put a reversible non-covalent interactions where then you can actually control the binding affinity through different the binding binding affinity. You can control the kinetics. And so we use a heterodimer, which is again designed in the lab, and then put that heterodimer here, and then let it basically assemble together. And so what we found is that so if you use covalent interactions, you have these hydrogels, and if you deform, it just stays as a deform. But if you take this non-covalent one, which has a rapid exchange, they can fall on and off. So they if you deform, it'll deform, but then quickly it comes back to the original whatever the thing. Yes, question.
What is the affinity of the heterodimer?
Yeah, so this it's been it was in nanomolar. There's on and off kinetics. It's not in my I can quickly look into, but I can tell you later. But there's different on and off kinetics. Yeah. So and then so it actually after deformation it goes back. So this kind of behavior in fluid, it's called also known as non-nuclear fluid, because they behave in a different way to different kind of stress and different kind of stimuli. And so then we actually measured the properties of this material, and we found that I'm not showing the data here, but we found that when these non-covalent assemblies are under rest, they actually behave like a fluid. Regular fluid. However, when you give them stress, they actually behave like a solid. And we have done all the rheology experiment to prove that. And then we also made this inside the cell. And again, we did the FRAP experiment, and where basically you make these condensates, and if you shine laser, it goes away, but then it comes back again to the normal shape. So so this we had a lot of fun doing all this. It's most of these are very exploratory, because we try to build things that can be controlled from outside, and which can also give different like fluidic properties within the intracellular space. And And so what I've shown is that we can build things with different fluidity. For example, we can have non-Newtonian fluidity, and also we can have freely diffuse assemblies, but also we can have very rigid. Yeah, question? Okay, so okay, yeah. So and then have a more stronger kind of assemblies, and it.
So how were you designing the actual disordered sequences? Like, did you one disordered sequence you used?
So this so it's a good question. A lot of people when I was there, a lot of people used to come like, oh, we want to design this intrinsically disordered protein. I mean, for intrinsically disordered, I think there's no I don't know if there's a design principle yet. And then the other thing is that because they are floppy, and you can just have like random glycine, serine, and things like that. But I don't think there is a design principle yet to design intrinsically disordered protein. Yeah, and so we stopped there, but then our collaborator who hold the first lab, they are now collaborating with David Baker's lab to basically explore the functional side of this this intracellular condensate, and how can you basically mobilize a certain like signaling molecules there, and how can you really use them for cellular engineering. So so I'll move on for my next. So these are all designed. My design, protein design, projects that I did at IPD. And then, so once I moved here to HMS, I really wanted to use all these proteins to actually really engineer cellular system where, like I said, my original idea was to, build this cell-cell interactions hub, signaling hub, where you can actually control cellular signaling. And so that's what I'm going to talk about now. And so one of the cell-cell interaction or the cellular signaling that that happens is a notch signaling. And notch signaling, and many of you might know that notch signaling is it requires two cells, and one cell basically express a notch receptor, and there can be 4 different notch receptors. And then the other cell the other cell is a sender cell which express notch ligands, and there again, the ZAG and then delta ligands, there at least 5 of those ligands. And so what happens is that when this the sender cell and the receiver cell interacts through this notch receptor and ligand, these cells actually pull each other. And when cells pull, there's an extracellular protease called ADAM. It cut it here, and then subsequently there's an intracellular protease, which is gamma secretase. It further cuts the intracellular domain of the notch receptor. That goes to the nucleus, and it activates many of the downstream genes. And so this notch signaling, which requires, first of all, the sender cell and receiver cell interaction, it requires the mechanical pooling. It actually controls almost all of the developmental fate decision in so like any early development, all the fate decision of the cells when cell differentiate, the notch signaling plays a very big role in controlling those fate decisions. And so so because here at George Daly's lab, because we work on hematopoiesis, and we are very interested in actually exploring this, the notch signaling in how it actually controls the hematopoiesis. So the hematopoiesis is basically the generation of blood cells in our body, right? And that begins very early during development. And so one thing happens during early development is that these hematopoietic stem cells, which are which come from this AGM region, and then later they move to bone marrow, and these hematopoietic stem cells are the one that will create entire blood cells in your entire life, your rest of your adult life. And so, and then again, this hematopoietic stem cell, at some point, they will go to thymus to become T cells, right? And they become all different kinds of cells, like B cells, dendritic cells, and all that. So all of those actually require that notch signaling, and this cell-cell interaction is important for their differentiation. Otherwise, you won't be able to have a hematopoietic stem cell or neither T cells, or you won't be here if there's no signaling, notch signaling. And so so because notch signaling is so important for all this cellular differentiation, when people try to make these cells in laboratory, in culture, then you must provide the NOT signaling. Otherwise, you won't be able to make any of these cells. You know, T cells, for example, and it's very popular disease for CAR T cells. If you want to make T cells in the lab, you must provide the NOT signaling there. And the way people provide NOT signaling is because it requires this mechanical pulling. So either people will provide this NOT ligand through another cell, it's called stromal cell, it's just like the Sander cell, or people would coat the plate with NOT ligand. So because like I said, it requires the mechanical pulling, otherwise you're not going to get it. So when I came here, so I had this idea, because this is again, this is cell-cell interaction, and it requires you know, like pulling between the cells. I thought the design protein could serve as what we call soluble NOT agonist. So you can have an oligomeric protein complex. You can just add this oligomeric you know, NOT complex to the cells, and if this oligomeric protein complex can bring two cells together, and if these cells, while you know, they're dancing around, if they can pull enough, they will then activate the NOT signaling. And so the whole goal was to build to develop basically a soluble NOT signal, which could be really useful for, you know, making like T cells in the you know, like large scale production of T cells for CAR T cells and things like that. So so how are we doing that? So the idea is very simple. We would take some of these design oligomeric proteins with you know, right arm lengths and things like that, and then we can actually we can affix this DLL4, which is a NOT ligand, to this protein complex through some kind of conjugation, like spike-tech, spike-etcher conjugation that I talked about. Then we can make this oligomeric the NOT ligand, which is DLL4, and so we can do that, and we wanted to have our a library of protein complexes in two configurations. One, we wanted to have this DLL4, which is a NOT ligand, in one side of the protein complex, or we wanted to have in the opposite side. So we wanted to have both cis and trans. And most likely that cis will basically bind to the same cell, so it will do a cis binding. And if you have a trans ligand, then it may be able to pull two cells together because they are, you know, pointing in opposite directions. So that's trans binding. So we explored a library of protein, and many of these actually, you know, we made in my previous previous work that I showed. And so we have both cis and trans binding ligands, scaffolds. So for example, here I have a dimer, and this is a homodimer. And as you can see that this is a trans homodimer. It's basically the red dot means that this is where you can install the DLL4. And so then you can also have, this is a C3, which is a trimeric protein. And this is because it's flat, so it can also act as a trans DLL4. We can also have a trimer where our DLL4 can be only in one side. So this is a cis configuration then. So likewise, we can have a hexamer with cis configuration versus we can have a hexamer, which is a dihedral symmetry, but it is a trans configuration. Likewise, we can have octamer, cis configuration, we can have an octamer with trans configuration. And then we can have larger cases, which will be also trans, because the large case is a, you know, rounding, so it can pull cells from both sides. So so then once we have this, and we wanted to test whether we can actually activate NOT in a reporter system, right? So again, the idea is that we have two cells, and we have this trans configuration oligomeric DLL complex, and that should be able to bring the cells together. And then we built in a reporter here. This is a U2OS cell lines, and we have a reporter which is citrin, which is a fluorescent protein. So if NOTs get activated, basically this GAL4 gets released, and it turns on the reporter genes down there. So we did screening, and this is the screening result. And so we basically use flow cytometry, because these are all fluorescent proteins turning on. So you can use flow cytometry to quantify how many cells are turning on. So here, if you look at the results, so there are some things very striking. First of all, C3, which has a kind of, you know, flat structure, therefore it's a trans configuration and has the highest NOTs activation, versus C2, which is, despite the fact that this is a trans, it has actually lower activation than C2. And then for the rest of the structures, all the structures that have cis configuration have very low activity. However, all the one that has trans has slightly higher Notch activation. However, this activation is less than C3. So it appears that the C3 exhibits the highest Notch activation and in a concentration dependent manner. And then this activation most likely appears to be preferred by trans configuration. Yes.
Do you have an explanation why the highest concentration at the 1 micromolar is less activating?
Yeah, so we actually we did a lot of experiments. So I don't have those slides here. So there is something called Hooke's effect, where what happens is that if you have a lot of the protein, then it just binds the cell and it coats the cell, and the cells are not interacting anymore. However, if you have the right concentration, so there is like the protein is bound there, but there is available ligand on the other cells, and they can actually come together. So we actually did a lot of Hooke's experiment there.
Why do you think you see that for the one symmetry but not for the others?
So the Hooke effect?
Yeah.
So that's also we did we do see Hooke effect for all the trans configuration. However, what may be happening, because why? So there is a question here that why this the other higher order symmetry is actually less than C3, right? I mean, they're still activating, but why less, right? So one of the things actually we are in the process of doing the experiment, but even from that Hooke effect, what we've learned is that so a lot of these higher order proteins they may have DLL4, which are, because there are so many, there might be some unbound DLL4. So you might actually waste some of those DLL4 because the concentration is so high. So that obviously I still, but from the Hooke effect, it looks like that's what it is happening. Yeah.
I was wondering what you're relying on to supply the mechanical force. Like you just assume cells are moving and then that's causing.
So for that's I'll come for that. For the adherent cells, because the cells are motile, they will constantly if you look at the HeLa cell one day and the next day you go back, you don't find it the same position, right? So they're moving around. So that is enough for the mechanical pooling. But for suspension cells, you need to actually you need to do a little bit of coupling, and then you need to give them some mechanical force. Yeah. So that was the flow cytometry data, and then this is this microscopic evidence to show that you have this turn on the fluorescent gene. So the question coming back to despite all this trans configuration, for example, C3 versus C2, right? And the data showed that C3 has highest activation versus the dimeric C2. Despite it was a trans configuration, it did not have enough activation. And why is that? And what extra thing you actually need for activation of the Notch pathway? And so in order to figure that out, we did this experiment. We basically tried to see the binding affinity of these oligomers to the cells. And so here we took U2OS Notch1 expressing cells, and then we took DLL4, which is a monomer. This is only the DLL4 ligands. And then we had a C2 DLL4, which is a dimeric trans configuration. And then we had C3 DLL4, which is a trimeric configuration. And so we did a titration, and we found that the C3 binds the highest, and C2 binds intermediate, and DLL4 alone hardly binds. So again, this is because the DLL4 has actually low micromolar affinity towards the Notch1 receptor. It's only like some 12 micromolar or something like that. And the concentration range that we are working with, it's kind of, like from micromolar to like nanomolar or picomolar. So the DLL4 alone, because of that, the binding is super, super low. And so as you have these oligomeric scaffolds and higher the symmetry, so you have basically this cooperative binding. It's binding more because they are all together, right? And so when we then look at these cells under microscope, what we saw is that when you have DLL4 alone, you see Notch1 receptor spread all across the cell. It's on the surface. And you don't see any DLL4 binding because of the weak binding. And however, when you put this dimeric DLL4, which is again in trans configuration, and it does bind to the cells, you see this tiny dots, puncta at the DLL4. And when you co-localize with Notch receptor, you do see this, at the cell-cell junction, you do see this, the clustering of receptors. However, you still see that the Notch1 spread all across the cell. But when you go to C3 DLL4, which is a stronger activator, and you start seeing the accumulation of this DLL4 in clusters between the cell-cell interaction junctions, and you also see the Notch receptor being totally clustered into wherever there is C3 DLL4. So yeah.
What about with regard to stromal cells? Is it as effective as whole culture stromal cells?
So for activation, you think? So actually, my initial days, very first experiment that I did, I actually tried with stromal as well as a control. So the problem with stromal is that, so there are multiple things. First of all, whether all the cells are getting signal or not, right? So depending on your cell density, so maybe like not 100% cells are going to like it depends on how many contacts, right? So, but however here, as long as you have right cell density and as long as you're providing the ligand, so it's literally every cell gets, not 100% cells, but most of the cell will get. So it all depends, even for stromal cell, like I said, depending on how much cells there are. And like, not always that there would be a stromal cell and a receiver cell close to each other, and that will depend on the cell density. Yeah.
Very question. This is very clear from here between C2 and C3 and DLL4. But I was wondering, what is the endogenous notch effect is? Like, did you knock down the endogenous notch?
So yeah, so here that endogenous notch was totally knocked out, and this has no notch one. So it is totally knocked out, not knocked down, knocked out. And then for activation, we do not have the endogenous pathway. We do not have the intracellular domain of notch. We actually put a GAL4, and then that GAL4 will then, when we have a reporter, it will actually turn on a new GAL4 recognizing thing. So we're not doing anything with endogenous system here. Yeah.
Do you think you may have additional interactions between the deltas and the notches, like more of the crisscrossing? And that's kind of, because you're artificially bringing things, I guess, together, where normally, I guess the model is normally like one notch, one delta.
So
bring them together, you get like a combination.
Yes, I will come to that point. It's in somewhere in the, I think the next slide. All right. So yeah.
Yeah, actually, I have a question. So in your first panel, I don't see any DLL4, but I do see notch, right?
Yes.
So I was wondering, what happens if you culture cells with just C2 and C3 without the DLL4?
So I did not do a microscopy for that. However, I have done enough activation with those controls, so I don't see any activation. So I presumably it doesn't bind. So in here you also don't see DLL4. Like I said, it has only like 12 micromolar binding affinity or something like that, whereas I'm using here 10 nanomolar here. So this is extremely weak binding, and you don't see that. So coming back to your questions, like what is the is it the so this, by the way, we call this notch synapse here, and notch synapse is a very new word. When we are doing this work, we thought we are the first one to sort of find that notch synapse, and we're going to coin that word. But again, one of our collaborators actually, they also found a very fundamental understanding how notch synapse work, and so they just published about a couple of days ago a paper. So here. And so what they found, this is actually work from Steve Blacklow's lab here at HMS BCMP department. What they found is that if you have a receiver cell and the sender cells, and when they interact, they actually, they cluster the receptors, both the notch receptors as well as the ligands. And so they say, they coined this notch synapse basically. And so what is happening in our case is, again, it's forming the synapse, and it's only the degree of synapse may actually dictate how much activation you are going to provide. And that can be, again, like in development, there's like no precise, well, we don't understand yet, but there's no precise amount of signal amplitude required for particular thing. For every different development, there might be a different requirement, like, how much signal, and that might be dictated by the degree of notch synapse formation here. So, yes.
So based on that, do you think you need like mechanical force, or can you just cluster?
No, clustering is not enough. We have done some experiments to that too. You need both. So it's a combination. So from here, I just want to conclude one sentence is that it's a combination of the ligand orientation in our case, and then the receptor clustering, degree of receptor clustering, and also the degree of synapse formation, as well as the strength of binding. So it's a combination of all that. So if you have all the things right, then you will get the maximum. Again, you'll get some activation, but not as high as that you want to see. Again, that high may not be necessary for your whatever you're trying to do. It all depends on the what kind of cell type you are working. So anyway, so then so those were all adherent cells, U2OS cells, and then we moved to suspension cells. This is K562 cells, and when you have no C3DLL4, then again you see that Notch receptor all spread all across nicely. However, when you put this C3DLL4, then your receptors are being clustered at the cell junction here, and this is actually my favorite picture that I took. It was like just a regular microscope in the lab, but you can see very clearly that the cell junctions and the synapse, the Notch synapse that's forming here, and then again it basically it proves that what we are seeing is the synapse, and we have done the activation for here. So this is a suspension cell. It forms actually larger clumps over time. We have done a lot of this. Hooke's effect experiment. What is the how much ligand is required and things like that, and at what ligand concentration this cell-cell interaction is efficient. I'm not showing those data, but so we found it out, and then because it forms large clusters, sometimes you also have to apply mechanical kind of shaking in order to break this so that it not gets the question.
A question from the folks online. Well, maybe first to say is the questions in the room are sometimes a little quiet depending on who's asking. So if you wouldn't mind just repeat the questions when someone in the room.
Oh, I see. Okay, sure. Yeah.
But there's a question from online for the transbinder. Why do you think they don't work as well as the C3 cis binder? Will removing one side of the binding recover the activation?
So the question is why other transbinder don't work as well as C3. So that's we have some kind of evidences and some hypotheses. I still am doing an experiment to actually find out the actual reason. So one of the reason may be because the higher order oligomers, it has so many DLL4s. Some of those DLL4s might be actually wasted, and then, for example, in the icosahedron case, there's so many, and they're not all are engaged. So the total molarity actually drops because of that, like the functional molarity of DLL4. And so, but, we did clustering experiment to show that that is the case. However, I still need to find out exactly what degree of binding is happening. So that's still underway. Yeah. So then we did activation here, and again did a lot of other work. We also tested this in different cell line with endogenous knots. For example, this is MD/MB cell line. This is SVZA cell. These are all cells that have endogenous knot system. There is activation. You can see that the downstream of knot has one, HA1, and multiple different target genes of knot. You can see then activation. Again, the activation of these genes are not very great. And if you look at the literature, you will see maximum 2-folds or 3-folds. You don't see a huge increase in the downstream gene expressions. You also do see some through Western blot. You can also see the cleave knot 1. So then finally, because again we work in blood in the lab, we work in hematopoiesis, and we have all this T-cell differentiation platform. So and because T-cells it is indispensable for T-cell development, the Notch is indispensable. So we thought, now if we put this C3DLL4, can we make T-cells now? And so we put into platform, and then so ideally it's a very long differentiation procedure. However, but just for simplicity, I just so I'm showing you that we take cells from cord blood, and cord blood has all the hematopoietic basically precursor cells, and then we differentiate those cells step by step to make T cells. It's multiple weeks long protocol, and then at some point we actually just squirt this soluble knots. Again, we did a screening which construct can do better, and then we found that the C3DLL4, which work very well for us, it can make T cells quite efficiently. It was still not as great as the 2D format that people have been using, but then again it requires some kind of optimization and finding the right concentration and things like that. So and then finally, because we want to use this in a 3D cellular production platform where we can actually make these T cells in a bioreactor, and because T cells have so much demand in CAR T cells, and so what we did was that we took iPSC cells and we differentiated these iPSC cells all the way to pro T cells, and then moved them to a 3D bioreactor, and this is where we use our C2DLL4. And the data here shows that if you have no C2DLL or no DLL4 at all, then obviously you don't get any T cells. I mean, T cell means CD4, CD8 double positive T cells. However, if you have the C2DLL4, you get a significant double positive T cells. And then this is comparing with 2D format that people do. It's not, again, it's not, you know, again, this is, I mean, it's difficult to compare, but as you can see that you have good double positive T cells. And then when you look at the other markers like CD3 and TCR alpha beta for functional T cells, and you can see those markers as well. So so this is all great. We can engineer cell interactions. We can bring cells together. We can have NOTS activation going on. We can apply this for T cell production and things like that. However, the NOTS, like, you know, I said at the beginning that NOTS signaling is not that simple, because it has a repertoire of NOTS ligand and it has a repertoire of receptors. There are 4 NOTS receptors and 5 ligands. And what happens is that during this development, they don't use, you know, what happens is that they actually sometimes work combined. So there's a combinatorial effect of all these ligands and receptors, just like our, you know, our taste receptors. You know, you eat something and the same set of receptors work, but then when you eat something else, it's just a difference in signal, but it's the same set of receptors. So for development, it's the same thing for NOTS signaling for all different kind of, you know, neurons or you know, vascular endothelial cells or, you know, blood lineage. They all have this combinatorial effect of NOTS receptor ligands. So, for example, in the bone marrow, when these hematopoietic stem cells they're differentiated, there are, you know, multiple NOTS receptors and ligands involved in this process. We thought, what if actually we present this in a combinatorial way? If we present all these ligands, can we then control the intensity and amplitude of signaling, which would be then precise for particular differentiation, like let's say for blood or T cells or B cell. B cell also requires at some point, or even dendritic cells, right? And or even neurons and things like that. Can we then control that in a precise, the amplitude of the signal? And so then, so we have all the ligands and we have the receptors, and they're all, you know, giving signals. And so we wanted to use in a combinatorial way. So in order to do that, you need to have now different kind of protein scaffolds, because you have to accommodate multiple ligands, right? And so, for example, if we have 4 NOTS ligands here, we need to have at least a hetero, you know, tetramer in order to accommodate all of them. Because so far I'm only using a homo trimer to accommodate only one kind of ligand. So now I need a hetero scaffolds, and so that, you know, I can conjugate and then present in a combinatorial way. I can control what I want to present and how much of it. Like, do I want more of, you know, delta 4 or delta 1 or vice versa? So because this requires hetero oligomeric platform, and so at the beginning, I think I mentioned that making hetero oligomers was very challenging, and especially when I was there, you know, there was a Sherry Bermeo who was trying to make you know, heterotrimer, and she had really struggling to make this heterotrimer for many reasons, but it was a you know, quite difficult problem. But however, with recent you know, diffusion, and Sergey is sitting there, who is also part of this work with diffusion, you know, I run diffusion protocol multiple times. It's just you know, the success is so great. And so this is a very new protocol for designing proteins, especially for backbone generation, which is based on the regular diffusion, you know, like the image diffusion method. And so using that protocols, you can actually build backbones and then design site-set sequence using other protocols like, you know, MPNN, which is also, I think only last year it came out. And then finally, you can use things like AlphaFold. And speaking about AlphaFold, AlphaFold 3 is there, and there's a big noise today. And I was worried that people are not going to be interested in my stuff because there's AlphaFold 3. So anyway, so you can use AlphaFold 2 to then look at this and see whether you can actually your prediction is accurate, and you can very quickly you can filter things amazingly. And I'm actually very impressed by this program. And so yeah, I'll come back to you. But Sergey, big thank you for you, because you set up a lot of this Google Colab thing and both Colab design, and I've been using it a lot. Yeah, sure. Yeah. Yes.
So what do you look for when you run confidence?
Yeah, so I look, and so I've been also talking to some of the people who are here. So like Helen, for example. So different people have different preference, but you obviously you look for. Huh? Oh, sorry. The question is what do you look for when you use AlphaFold? Like what kind of scores? So there are multiple metrics, and one of the things I was told by one of the person is that look for RMSD. RMSD is one of the very the key things you want to have within 1.5 Armstrong or something like that. There are other scores. Then you can use PAE and things like that, or all the key scores. But however, you can optimize, and it also depends on how big of the protein that you are making. So if you're making like really large protein, like oligomer of big size, then your score drops. So it's all very depends what you are trying to make. If you're making like smaller things, then obviously all of your scores would be shooting very high. For example, RMSD, some of this design that I'm doing, it's like really below Armstrong, 1 Armstrong RMSD. Yeah. So anyway, so you can use this protocol, and I've been having some fun, especially I run before I go to sleep. It's very fun. So I actually managed to have some very test run, and these are some heterotrimer, for example. It looks beautiful to me. I mean, and this is a heterotrimer where you have this alpha helix and you have the beta strand at middle, and it just looks very amazing. And also I designed some heterotetramer here, and again, they looks really good and with great scores that I got. I only did this in silico so far. I have not tested. Maybe soon I will test some designs in the laboratory and try to make that. But if I can make this, it would be amazing, because then I can I have all these knots ligands, and I can put all them together, and I can also optimize based on trans versus cis, and I can filter based on what I want and what I don't want, things like that. So this is going to be my final slide before the summary. Yes, sure.
So is there a reason other than that you just haven't gotten into it yet, that you haven't designed and tested those?
It's only, I think, last week or so I designed it, and I'm trying to submit the main paper, which is almost there. And so yeah, I will at some point I will definitely do that. No, yeah. So then I'm just trying to get back to what I started with. I wanted to build a signaling hub that is so complicated, but at the same time it provides me to control over how I can control different signaling molecules, put them together, and do really very interesting. I can ask interesting questions. I can use them for the immune cell interactions and things like that in immunotherapy. So now when I started, I said we did not have really good scaffolds, like for example heterotrimer and even heterodimer that can have like this on and off different affinity and things like that. But I think with these new tools like diffusion, I'm very excited actually to now we can build more of this, and then maybe this is going to be possible very soon. And if we can make that, and obviously we can use for all today I talked about obviously not signaling, but you can use it for all different kind of cell-cell interactions that you can control. So to summarize finally, so what I talked about is today, using helix fuse and worms to build, higher order protein complexes, and then using that platform, then we build this intracellular protein condensates. That again, we didn't go far for doing some really cool, signaling there, but we showed that this can be done. We can control the mechanics and things like that, and the other people are now exploring that how we can actually, use it for cellular engineering. And then finally, I talked about engineering the cell-cell interactions, how and giving an example in not signaling system, and I talked about how we can use that for designing cells like T cells, and I think this is going to be very exciting because there's so many different kind of cell-cell interactions. So I just want to thank people. This has been like a long journey, and started with David Baker's lab. A lot of people contributed: Ion Xia, Will Schaffler, Natasha, Tize, and Una, and other people. Justin helped me doing all the FRAP experiments, and then Helen, who is also in the diffusion work now. I collaborated with her. She has been great help. And then I also collaborated with DeForest Lab, where they do all the rheology work, and Rose and Nicole from there. And then here at HMS, George and Ran Jing, who is expert in T-cell differentiation, and then lots of other people. And then also at Trista's lab. Then Steve Blacklow's lab is the NOTS lab here, and I collaborated with Steve. And then people, Arvin Landel, who is at Karolinska, and then lots of other people, friends and families, and Rajesh, who's sitting there. Yeah, so that's all. Thank you so much for your attention. I'll be very happy to take more questions. Yes.
Great talk. I was wondering if you've tried your system in the syn-notch style system. Like, is there anything particularly special about the delta 4 JAG1 ligands?
Yeah.
Let's say you use a GFP nanobody pair or something. Would you lose some of the efficacy the efficacy?
So the question is, did we use this in syn-notch system? So the definition of syn-notch is a little bit vague because, like it's basically, you have the entire notch signaling, and then you have different parts of the notch signaling. You're basically re-engineering, and I think that's what it's supposed to mean, syn-notch. But if we talk about very typical like Wendell Lim style syn-notch that he uses for CAR T cells, then we have not used that for that, but I just want to remind you is that the reporter system that I built, that is also a syn-notch actually in one way, because we have notch receptor there, and then we have GAL4 inside. So it is kind of syn-notch. The interaction, extracellular interaction domain.
The more that the interaction domain.
Yeah, no, we have
is there
not.
anything extra going on there that leads to that synapse formation?
Yeah, we have not explored that. We have not explored the extracellular, antibody, whatever, SCFP or any kind of this. We have not. Yeah.
I was struck by your last slide that was it was notable that your last slide, which was produced years after your first slide, you had the same motifs of alpha helices and beta sheets, but this came from RF diffusion, the most modern. Those programs only know the same stuff you knew back then. There's like hundreds of other folds that proteins use, but they don't appear in any of your designs. What is it about these programs that converge so strongly on the things that your intuition led you to? Is it because it was trained by Baker Lab people and they only know what they put in?
That's a wonderful question. The question here is that why do you see with this diffusion model, we see all different kind of folds, but however in the old days mostly you see alpha helices and things like that. So because the old style of designing protein was, Sergey is here who is more expert than me, but the old style was that it is all based on physical interactions. Like it's all, we call it physical. Rosetta is based on basically all the physical interactions. The new styles are all machine learning, and they're like,
it looks just the same. It's all alpha helix and beta. Your new design is no different than the others. That's the same secondary
structures. Yeah, so the new one actually has something more, and you will see a lot more alpha helix and beta strands together. In old style, because you build alpha helix using some, we used to call like trig parameters or things like that. So it is exclusively you're trying to build only some, I mean, of course, there are other people who try to build both of that, but very less successful. So with new ones, I think it accommodates more kind of different secondary structures than the old style.
Went from 1 to 2.
Well, I mean, secondary structures, I mean, you have only alpha helix and beta strands, but how you can do is, I think it's a different story.
The machine learning methods learn patterns, and secondary structure elements have repeating patterns that obviously pop up and are more dominant. And in general, like AlphaFold predicts secondary structure way better than loops, unless there's like, they can cheat and use contact maps, like multiple sequence alignments, right? But like raw loop prediction doesn't work very well. That's why it doesn't really predict antibody loops very well. So it's just secondary structure works well. Use machine learning and the loops still don't work that great. And so that's why you see design structures, even with diffusion modeling or AlphaFold hallucination, still highly enriched in secondary structure elements. And you don't see a ton of like long loopy stuff.
The ones that make it on the slide are also the ones with good scores.
Yeah.
When you design something computationally, what is the actual success rate that the
Yeah, so again, I have not done any, I have not tried to characterize anything in the lab. So I do not know what is going to be in my hand. However, this diffusion paper, depending on what kind of structures, there are different degrees of success rate, and they call it in silico versus, and then.
just from your design for the notch, like are you those the DLL?
So those, okay, so that is old style, and in old days we used to do using that regular Rosetta, and so that used to be, we used to filter a lot. And so when we filter, again, it depends on the structure. If it is like icosahedron K is large, then it might be like from like 2% to 10% or something like that, depending on your thing. However, if it is like small scaffolds, like trimer or dimer, the success rate might be very high. So yeah.
When you have these failed ones, when you have the ones that are not working, like not the failures, but the other ones, where does that data go so that other people can learn from it?
That's a very good question. So that is a very good question. In old days, we would only report in the paper that out of 200 design we designed. I don't think we actually put all the failed one, right? Do we? We don't, right?
There were several attempts.
I see. Okay.
Gather all the failures.
I see. Okay. I see. Okay.
They're in some forgotten corner of the Baker Lab servers.
I see. Okay. Okay. I'm not aware of that.
Maybe they want others to have fun too. Sorry, no question. In figuring out what fails, what doesn't.
There's a conversation going on here, so I'm trying my best here.
I'll also say things can fail for a lot of reasons. And so it's really, when something succeeds, it's like, oh, great, it's good. But if something fails, to put it in like a fail bucket so that people train on it and believe that it's failed, it's a lot more work to dig on that. And yeah, not a good way to gather all the stuff and share it.
I think somebody, quite a while ago, I attended this talk by Francis Arnold, who talked about designing enzymes based on all the failed data, basically. So I think that could be interesting. Like what if, like machine learning is supposed to be learning from not only from success but from failure as well, right? So I think that's a good idea.
So what about a partial failure? Or if it doesn't fail all the way, if it doesn't work quite well enough, or it's weird?
And then another thing I want to just mention maybe here. So for most of the Rosetta design, I briefly mentioned that these proteins are the one that works, they're so stable. You boil at 90 degrees Celsius, nothing happens. And so that's also because you design in a way that they are like super stable and really like folds well or something like that. So there might be a lot of other possible intermediate level, whatever folding, and that might just work. And maybe we did not look at those, right? So.
Awesome.
Yeah.
Rubul, thank you so much for a really fantastic seminar. Yeah, we have quite a lot of pizza and beer left. No one else needs the room for the rest of the night, so feel free, hang out, chat, socialize, ask Rubul some more questions.
If you have any more questions.
And we'll see you across the river at MIT next month.